The next speaker is Dr. Benventri. See, I messed it up. I'm so sorry. You should hear my Spanish. He is Chief of the Renal Unit and Director of the Bioengineering Division at Brigham and Women's Hospital, and has had a long-standing interest in various aspects of cellular injury and repair mechanisms in the kidney, with special emphasis on the role of inflammation, biomarkers, and stem cells. He has established the origin of the epithelial cells that repair the kidney after injury as differentiated surviving proximal tubular cells. I got that, guys. He was the first to describe the role of proximal tubular and cell cycle arrest in the maladaptive fibrosis that can occur after severe injury, leading to chronic kidney disease. He discovered and characterized kidney injury molecule 1 as the most highly upregulated protein of proximal tubular after injury to the kidney of various types. He has also created the iPS cells from patients with adult onset PKD and is now working on ways to differentiate cells down the kidney lineage. Dr. Benventri.
Well, thank you. This is all about our patient, and clearly the motivation is there. I'm going to talk about an initiative that we're all very excited about. It's extremely ambitious. On the other hand, the time is right. Dialysis has been with us for 70 years, and really we owe it to our patients to have made more progress, and we will make more progress going forward. And this talk is primarily about organization. It's not so much about technology. And if I can get the first slide, will I be able to see? Okay. Okay, it's on. So now, okay, good. Now I can see. Okay, so now it says dialysis has changed little over the last 50 years. And again, it's been with us for 70 years, but obviously early on there was a lot of change, and it's been very important. It's kept an enormous number of people alive, and it's been certainly something that patients have benefited from. But compare the lack of innovation to the incredibly disruptive innovation that's occurred in other fields over these years. Compare it also to the cures that we have seen in things like childhood leukemias and a whole host of other disease processes. Our dialysis patients continue to have poor clinical outcomes. Their quality of life is poor. The cost of care, as you've heard already a little bit, but you already know, is enormous. And kidney transplantation is really not the solution. It's really insufficient. The organs are insufficient to deal with the growing demand. So how do we facilitate novel approaches to renal replacement therapy? And I think an important part of what the Kidney Health Initiative decided, and the leadership, even before I was involved, was to actually make the statement that in fact we can make progress, and we have to move forward. We have to move in a timely fashion, and we can't just say, well, it's just too hard. Okay, and the time is right for innovation. This is not a technical talk, and I think John Himmelfarb will talk a little bit more tomorrow about technologies. But, you know, there's action in this field. Artificial kidney, you've heard about it, wearable kidney. You see in the middle there this device that's sort of hooked on to the blood vessels in a rendition of Shruvo-Roy and Dr. Fussell. And then to the right, you see a system of printing whereby you see flowing through this system, where you can imagine now putting the cells that replace the proximal tubule or the tubules of the kidney lining these devices. This is in work done by Dr. Jennifer Lewis at Harvard. And then in the bottom left-hand side of this slide, you see the organoids, these self-assembled structures that demonstrate that you can take stem cells and make kidney cells from them. And then in the middle and the bottom, you can see that you can basically think about putting cell layers together to try to reproduce the filtering unit of the kidney. But there are many other, there's sensor technologies that are not reflected on this slide. There's an enormous amount of material science, and as has already been mentioned, we really have only scratched the surface in engineering advances and miniaturization advances. Okay, so what is road mapping? And this is what we're talking about. So a roadmap basically outlines a logical time-based pathways for technology advances in a given industry or organization. Why? Why develop it? To motivate the right people to take aligned action, accelerate innovation and technology development, and encourage investment. Characteristics of an effective roadmap. To incorporate inputs of diverse and representative groups of stakeholders. An important part is to refrain from picking technology winners or losers, to encourage innovation and really leave everything open, basically. It's effective as both a planning and communication tool, must be clear and compelling, and it's a living document that is updated in response to technology advances and market changes. And you'll see on some of these slides this Nexite group on the bottom. And that's a group that's had a great deal of experience in developing roadmaps, and they've been helping us. This is something over the last few months, and they've really made help us make some very significant progress. Okay. All right, and this group has actually developed the roadmap in collaboration with others in that particular organization for the National Cell Manufacturing Consortium roadmap, the NIST Advanced Manufacturing Technology roadmap, and the SESME Accelerated Roadmap Development, with the last one basically to inform Clean Energy and Smart Manufacturing Innovation Institute 2018 initiatives. Okay, and our process is one that, again, has patient voice at the center, as it has been at this meeting and will continue to be, and is bringing together industry leaders, FDA regulators, NIH scientists, CMS payers, White House leaders, and academic experts to really come up with an innovative approach, or innovative approaches. This is the steering committee. And you can see it reflects the KHI integral involvement of the FDA. And what was mentioned, CDRH has been mentioned, but CBER has been very, very actively involved here in this initiative. We've divided up into 4 working groups: a patient advisory group chaired by David White, a mechanical group, so we've sort of arbitrarily, but it's been useful to deal with these innovative approaches and roadmap from the context of mechanical, cellular, and vascular. And Frank Hurst from the FDA is running the mechanical group. I'm running the cellular group, and the vascular group, Prabir, is running. These groups meet by conference every 2 weeks or so. And so the roadmap components, the future state, high-level goals the roadmap aims to achieve to improve patient quality of life, identifying the challenges. There are technical and market challenges, with market challenges defined here as fiscal, regulatory, patient challenges in terms of preference, for example. And the roadmap activities have to really address or overcome these challenges. And what are the solution design requirements? Well, if you think about what's necessary to replace a functional kidney, there are minimum requirements for any renal replacement solution by kidney functional area. So blood filtration, electrolyte homeostasis, filtrate drainage and connectivity, fluid regulation, toxin removal, secretion, and access in the context of vascular access or peritoneal catheter access and integrating with the blood circuit. Milestones have to be set up as measures of progress, and as Murray has already mentioned, there's a great deal of pressure to make these milestones in a time frame that are shorter rather than longer, but still to be consistent with reality in terms of what we can promise patients, or what we can realistically think could be done in a period of time. So we have short-term, over 3 years, mid-term, over 6 years or so, and long-term action, outcome-oriented activities with a level of specificity that could align with potential fundable projects, because there's clearly the notion that this roadmap could link well with funding organizations, whatever those organizations might be, in order to achieve these well-defined milestones. This was a roadmap diagram that we brought out at the ASN Kidney Week in 2017 in New Orleans. And I'm sorry, the print may be a little bit small, but essentially at the bottom left, and it's not as complex a roadmap as we will end up with, but it gives you sort of some idea of where we're going. And there are incremental dialysis improvements, and in each, starting in sort of maybe some low-hanging fruit. There's regulatory considerations and there's financial considerations that are present at every step of this roadmap, and for which we are well prepared with the makeup of the roadmap group. There's wearable or implantable notions, so wearable mechanical devices or wearable biohybrid devices or devices which are purely biological. And in the left-hand side there on the second row, there's the chimeras and xeno. So we're not leaving that out, in particular xenotransplantation, because we don't want to, again, rule out any possibility for advancement that will actually make patients' lives much better. And then at the top right, there's this regenerate the biological kidney. So another goal would be to see whether or not it's possible to actually regenerate the kidney itself in a biological direction. Okay, let's see. All right, so the focus areas and categorization for the research and development activities. On the left are the functional activities of the kidney, mechanical and cellular. So blood filtration, electrolyte homeostasis, fluid regulation, toxin removal and secretion, filtrate drainage. You have to, whatever we build, it has to drain someplace out of the body. And there has to be a vascular access or a peritoneal access, some kind of access to the body. The integrating technologies, there are cross-cutting technology adaptation, particularly technologies that are not unique and not really well established in renal, but can come from other fields. There's function and safety monitoring that has to be incorporated. On the mechanical side, if it was purely mechanical, there's a dialysate supply. On the cellular side, there's the whole biomaterials development. And then on the cellular side, there's also the xenotransplantation that we have to consider. And I'm going to show you just two examples of some of the technology piece of this. But if we deal with filtration, for example, and I'll read this, develop a cellular size-selective, non-clotting blood filter capable of 40 liters a day with X months, we haven't quite filled that in, of continuous performance that will freely pass electrolytes and non-protein bound toxins connected to the circulation with or without a pump. Okay? That's something that we're conceiving because, you know, we're people and we have biases, but it's not meant to be limiting. So that's one thing. And then that system can involve cells or not involve cells. So the second category is to develop a system that actually incorporates cells. The third category is to identify and generate the cell sources that would be like glomerular filtrating, the components of the filter, glomerular endothelial cells, mesangial cells, podocytes. And finally, demonstrate function in a full-scale animal model as an add-on kidney followed by support for survival in an anephric animal. So those are sort of, and if you look at the timeline, you know, what we've got, we have a lot of discussion about the timeline, and this is not final in any way, but we think some of these things are on a 3-year time frame if they're resourced well, and some of them are going to take a little longer. Okay, and then, sorry, just one other thing, and this also makes another point. When we normally think about electrolyte homeostasis, the kidney does a lot of things, you know, controls sodium, it controls the pH of the blood, it controls the calcium level, it obviously controls water. And there's this intricate, complex cellular network that does this. But if you turn it over to the mechanical team and say, you know, is there a way to replace this without any cells, then they'll come up with things like this. You know, develop online sensors to alert the users that sorbents, that is biomaterials that will attract certain toxins, for example, whether those sorbents need to be replaced because they're saturated. And develop regeneration systems that could have a size and weight characteristic to them. Develop blood systems and renal replacement therapies to monitor the large, so here's the sensor piece that could lead to symptoms or complications. So monitor what's going on. And finally, again, from a mechanical point of view, is it possible to develop pores, a system with pores and channels and ion-selective membranes capable of selective removal of electrolytes, sodium, potassium, calcium, etc. And again, most of us, when we think about replacing these functions, think about cellular solutions. But again, I just wanted to include this slide to indicate that there's a whole other approach with a whole other group of individuals that are thinking about it in an acellular way. And of course, there could be hybrid solutions. So this is the last slide. What are the next steps? Finish the development of the design criteria and the R&D activities and establish the milestones. Obtain scientific feedback at the upcoming ASIO meeting. Develop integrated strategic graphics so that we can convey this in a way that makes sense graphically to people to understand, patients and potential collaborators to help accomplish this. In August 2018, we'll present the draft roadmap for public comments. We welcome all suggestions for improvements at this stage as we move this along. It's hard work, and we likely are missing some things. We'll refine this based on community input, and there will be hopefully a good connection with KidneyX and potentially to utilize KidneyX alpha funding for some of the goals and accomplishing some of the milestones. And over the next year, add additional research and development areas such as xenotransplantation or anything else defined by public comment. So with that, I think that's the last slide. So thank you very much. And Murray is going to say a few words to correct the things I've said or amplify on them.
Thanks, Joe. So we've heard about the Kidney X that HHS has graciously helped us put together. This started all several years ago when the Centers for Devices at FDA recognized the importance of stimulating innovation in this area. And as we've also heard, it's needed and it's long overdue. Innovation in the renal space is grossly lacking. So we're trying to develop this roadmap to help to de-risk the development so that developers can understand what FDA might be interested in seeing. Developers can learn from patients what patients are interested in having. Our Centers for Devices, Centers for Medicare and Medicaid Services can help identify how these products can be circulated to patients and paid for. And we want to overlap this roadmap with the Kidney X so that it will really provide some sort of guidance. The roadmap isn't developed, it's just a straw man. We want all of your input, and as it's being finalized, developed, it will go to KHI and all of its members to provide feedback. We desperately need the engineering group, and we will pick that up at AIO this June and ask for more input. But what we're trying to do really is to do something very, very different in this field. If you were to ask Edison what we should do to have better light, well, most people would say, well, let's improve on candles. And if Edison tried to improve on candles, we would never have a light bulb. So we know that dialysis has been around and it's helpful, but we don't so much want to improve on dialysis. We want to consider dialysis the candle, and something else, which we don't yet know, and what it's going to look like, is going to be the light bulb. And the light bulb is more than just an imagination or more than just a dream. But we need to do it, and we need to do it now. And we need to do it quickly. There's an old African proverb that I like to think about, that if you want to go fast, go alone. But if you want to go far, go together. And if we can bring together all of the components, all of the expertise from all of the disparate sciences and engineering to add in their special knowledge and their special niche activities to this, like a Manhattan Project. You know, there was a war a few decades ago, and it was very, very difficult, but we needed to do something to end that, and we brought together all of the knowledge and all of the skills. And in a very short period of time, we were able to end that war very quickly with the Manhattan Project. That's what I see for this, and I see it mostly for Caleb. And if we always remember Caleb and his story, I hope that provides the stimulus for us to do this now while Caleb can benefit from it. Thank you.